Organizing Committee of the Amity Institute of Organic Agriculture, Amity University, Noida. I, Dr. Garima Thakur, Assistant Professor, extend a very warm welcome to all the distinguished speakers, respected faculty members, researchers, scholars, and participants to Day 5, Session 1 of our Faculty Development Program on Climate Resilient Agriculture and Allied Technologies for Net Zero and Socially Inclusive Framework. I would like to express our sincere gratitude to our visionary leadership for their unwavering support and encouragement. We are deeply thankful to our Honorable Founder President, Dr. Ashok K. Chauhan Sir, Honorable Chancellor, Dr. Atul Chauhan Sir, Honorable Vice Chancellor, Dr. Balwinder Shukla Ma'am, Director General, Amity Food and Agriculture Foundation and Chancellor, Dr. Nutan Kaushik Ma'am, and Director, Amity Institute of Organic Agriculture, Dr. Doily Sanyal Ma'am, whose guidance and commitment have made this program possible. As we reach the final day of this enriching FDP, we have had the opportunity to explore diverse dimensions of climate resilient agriculture, ranging from climate smart farming systems and resource management to precision agriculture, artificial intelligence, genomics, and genome engineering. Today's theme, current scenario in plant breeding and genetics for climate smart agriculture, is particularly significant because plant breeding and genetics form the foundation for developing crop varieties that can withstand climatic stresses while ensuring food and nutritional security. Advances in genomics, genome sequencing, and modern breeding technologies are opening new avenues for creating resilient, productive, and nutritious crops for the future. So we are privileged to begin today's session with an eminent scientist and visionary leader in the field of genetics and plant breeding, Dr. Velu Govindan, Principal Scientist, Wheat Breeding, International Maize and Wheat Improvement Center, CIMMYT, Mexico. Dr. Govindan is an internationally acclaimed wheat breeder and crop scientist recognized for his pioneering contributions to biofortification, climate resilient wheat breeding, and accelerated genetic improvement in cereals. He currently serves as principal scientist, wheat breeding at the International Maize and Wheat Improvement Center, CIMMYT, Mexico, where he leads global wheat breeding pipelines and zinc mainstreaming programs aimed at enhancing nutritional security and climate resilience. Dr. Govindan completed his doctoral research in pearl millet breeding at International Crops Research Institute for the semi-arid tropics ICRISAT during 2003 to 2006. He has extensive experience in plant breeding, genetics, and molecular breeding, having worked on pearl millet, sorghum, and wheat improvement programs across Asia and Africa. Before joining CIMMYT, he served as sorghum breeder at ICRISAT, Bulawayo, Zimbabwe, where he contributed to FAO-supported sorghum seed multiplication programs for southern African countries. Since joining CIMMYT in 2009, Dr. Govindan has played a transformative role in developing micronutrient-dense wheat varieties with superior agronomic performance, industrial grain quality, and resilience to heat, drought, rust, and wheat blast. His leadership in the Harvest Plus wheat breeding program has facilitated the rapid adoption of zinc-enriched wheat varieties across South Asia, Sub-Saharan Africa, and other wheat-growing regions. His work strongly integrates genomics-assisted breeding, speed breeding, molecular marker technologies, and climate-smart agricultural approaches to accelerate genetic gains in wheat. His contributions towards biofortification and climate-resilient agriculture continue to support global food systems challenged by climate change, water scarcity, and hidden hunger. Through his scientific leadership, innovation, and collaborative approach, Dr. Velu Govindan remains a leading figure in global wheat improvement and sustainable agriculture research. So with these introductory remarks, I now invite Dr. Velu Govindan to deliver his presentation.
Audience, please wait. He's encountering some technical difficulties there. So I'm in contact with him. We are in conversation. So he's trying to enter. Please wait a few minutes.
Karthikeyan sir, you are not audible.
Wait. So
Yeah, he was audible, Dr. Chatra.
maybe you can unmute in your computer. He is facing some technical difficulties. He said he's not able to sign in. Just I'm trying to resolve.
No issue. He is at Mexico at this time.
Yeah, no, he is in front of computer to present, but having some technical issues. It will be solved in few minutes.
Meanwhile, we may discuss if any participant.
Even Professor Porchedu, if you interact with anything, we can join. Professor Porchedu.
We may request to Dr. Porchedu, other participants also,
Yes,
a kind of roundtable
participants, they can
discussion.
tell their feedback, opinion, and all,
Right.
how they can.
Uh, the second speaker, if it is possible, Dr. K, you may invite him.
Yes, Bain, sir.
If Dr. Valu is not at par, then we may request to Dr. Wayne.
We can wait, sir.
Certainly.
He is nearby in front of computer, but some technical things.
Dr. Shalu, I see you. If you have any suggestion, we may discuss over here meanwhile.
Um, Dr. Velugovindan has already joined, so we will be delivering a lecture shortly.
Yes, we see Dr. Karthik. Dr. Valu has joined, certainly. Thank you, sir.
Sir, good morning.
He should be unmute at his.
Velu, sir. Uh,
No, can you hear me, Karthik?
Yes, sir.
Yes, sir.
Very much. You are audible.
All right.
to us. Yeah.
Yes. My sincere apologies for this
Oh.
hiccup. Um,
Okay, sir. Yeah, thank you so much. I think you encountered some technical issues. Now it is resolved.
Yes.
So please kindly start, sir, your lecture.
Yes. First of all, thanks to Karthik and the Amity University administration for giving me this opportunity to share some experience on wheat improvement at CIMIT. As you all know, CIMIT is an international organization working on wheat and maize and climate related aspects, as well as now we are also working on the dryland crops like minor millets and other crops. So the title of my talk is Wheat Breeding for Global Impact, Advancing Calorific and Nutritional Security with Climate Resilience. So I have been with CIMIT Mexico over the last 17 years, and before that I was with the CRISAT in Hyderabad, and also shortly I was
Sir, actually, sorry to say, your introduction done. Our faculty, Dr. Garima, presented about your introduction already.
okay, all
Because
right.
yeah, we were
Okay, because time constraint, I will go with the presentation.
this one.
Yeah, so if you could see, CIMIT is the largest public sector wheat breeding program globally, and if you could see, the green revolution has started from here, and we do have good partnership with the national programs, including India, and so that contribution is really a remarkable achievement in terms of food production in India, not only in wheat but also in rice and other crops. So over the last few years, our genetic gain is about 1.5% to 2% per year. That is our average genetic gain.
Sir, please share your slide, I think. Maybe.
Oh, I'm sharing. You're not seeing?
Sir, DK sir, are you seeing the slide?
Yeah.
Okay, okay. Okay, sir. Okay.
Yeah. So there's a strong opportunity with this growing public and private sector seed sector in India. And previously, there is not much interest on wheat, but private sector, seed sector. But nowadays, there's a growing interest among these private companies. So we do provide the improved wheat germplasm at free of cost to all the public sector, and we do may have some charges for the private sector organizations in India. And if you look at the impact over the last 10 years, more than 300 wheat varieties have been released directly from CIMIT selections. It was sent to across different countries, and including India. So on average, if you could see, the CIMIT derived varieties grown over 40 million hectares, which is about more than 20% of the global wheat area. And for example, for in India, out of the top 10 wheat varieties grown, and 8 of them are derived directly from CIMIT. So that's a great contribution from our breeding programs. For example, more than 50% area is almost covered by CIMIT derived varieties. The same impact in other countries as well. So the yield gain is very important because of population growth, um, and the consumption also increasing, and the other global climate change issues. So despite all this, this is good, healthy gain in terms of wheat production in India. For example, this is like a kind of analysis done for every 10 years. So you can see in the early days from 1960s to until late, uh, almost 2000, the genetic gain is about 2 to 4%. But recently it has been declining, or making slow pace, because making gain on top of the um, high yielding varieties is complex. It's not easy. So I mean, the low hanging fruits are already harvested. So we are working now to improve. Further increase is bit difficult, but still we are making good progress. So where we originate, so if you could see Mexico, we do have 2 breeding hubs. One is in Ciudad Obregón, in northern part of Mexico, where do we have all our yield trials and segregating populations. We select for different stress, because this is kind of a desert environment. We don't have much rainfall, so only irrigated, so we can able to manipulate or control simulate different kind of environments by planting early or late, or depending on amount of irrigation we give. On contrary, we grow the summer season nurseries in Toluca, which is close to Mexico City, Here we expose our segregating population for different biotic stress like diseases, yellow rust, septoria, and fusarium. So this is kind of the shuttle breeding between these two stations, and also within same year we can able to grow into two seasons. So that is kind of speed breeding. But now we have made a lot of other improvements in breeding program. In addition to the Mexico hub, we do have partnership with many different partners, including in India. I will talk about that. We do have the BISA sites where Karthik was engaged in few years with us. We do have 3 stations in India, and also we do have other centers like in Nanjing in China, and we do have Joro, Kenya, and Ethiopia, and also Turkey. So there are different hubs. So once the advanced lines have been developed, we send these distribute for different phenotyping. It can be for yield phenotyping, it can for disease phenotyping. Then we get the data, and also the partners they use make use of this material for their breeding program. Yeah, so this is I already mentioned about the shuttle breeding. We advance material between these two stations, and our breeding cycle used to be like 4 to 5 years from crossing to the yield trials, about 4 to 5 years, and then the recycling happened after 5 years. This is kind of not acceptable by the partners or the funders because it's quite lengthy process. The breeding is a lengthy process. To develop new varieties, it takes quite a lot of time. So we have made a few changes recently. I'm going to talk about that. But what we target, so obviously the climate resilience. So by exposing our segregating populations to contrasting environments brings the stability factor and the productivity gain. So we select under optimal and stressed environments, make gains for yield traits. And also input is efficiency. What I mean here is we try to make selections for low nitrogen, or we call biological nitrification inhibition. I will explain a bit later. So we are working with that on using BNI, uh, using the microbiomes to increase the nitrogen losses. And of course the nutritional quality and end use quality, because whatever we do at the end, farmers and consumers, they have to use our wheat varieties. So we make sure the wheat varieties we develop, they do have better nutritional quality like high zinc, iron, as well as better processing quality like chapati making or chapati quality, and all these we do consider. So if you look at the Breeder's equation, so the genetic gain is directly proportional to the selection intensity and selection accuracy and the genetic variants we have in our breeding population, and inversely related to the breeding cycle time. So the selection intensity and accuracy we can improve certain extent, but it also involves a lot of additional investments. I will talk about that. But on the other side, if we could reduce the breeding cycle from 6 to 3 years, perhaps we can cut down the number of years, and then we can make faster recycling, and the genetic gain is much higher. So for that purpose, we have established this the speed breeding greenhouse based facility in Toluca station, uh, where we grow large number of the generations, early generations in the greenhouse, and the later generation we expose them to the field. So this we have optimized the 3 year breeding cycle uh, instead of 6 year or 5.5 years of breeding cycle, which used to be the case. If you could see in detail here, from crossing to the stage 1 yield trials, it's in just 3 years. This is a remarkable achievement because it used to take a lot more years. Now we have optimized this. Within 3 years, we can able to recycle our new parents. While doing so, and also once after 3 years we recycle, but also we go for stage 2 trials across different target environments. That's where we said large number of trials. If you could see here in this map, we send the stage 2 lines from Mexico all the way to these red dots are between Pakistan and India. This is the northwestern plains of India and also northeastern plains of India and parts of Nepal and Bangladesh and also central India. So this is the target. Three distinct environments. So we do send material and we phenotype and we get the data at the end of fourth year and then we still make use of the data. So by year 5, using all this data, we constitute the international trial, which I mentioned. So that is distributed across many different countries, not only in South Asia, but also many different countries. You can see here all these red dots in this global map is individual shipment we do every year to different partners. We share our Elite Gem Blossom. Yeah, so this is our 3-year population improvement scheme. I don't want to go in detail. This is with the numbers, but it is the same thing. So, and we are also trying to optimize 2-year plan that is in pipeline because on kind of piloting basis, we are trying this 2-year improvement cycle of recycling faster method also being optimized. So we don't have data yet, but we are in the process of optimizing this bidding scheme. So what is unique about the semi-wheat program is, as I mentioned, this Obregon phenotyping is quite unique. Hardly there is no rainfall during the crop season. You can able to simulate different kind of stress and also optimal environment, also different planting types. So this gives robust phenotypic data from within one location. So from the stage 1, we generate a lot of this data, and then we make the short list, and then the stage 2 lines goes to all these partners in different countries, and then they make selections, and then some of the partners they make use of these lines in their bidding program, and some of them they get the data to build these prediction models for the genomic selection. So what we have achieved, so the 1% yield gain, it doesn't look that great, but if you look at 40 million hectares, 1% is quite a lot of wheat. So we are making slow incremental gains in terms of yield potential, despite all this climate change, especially if you could see this year, the northern part of India experienced quite heavy heat stress. So that has pushed wheat production bit down, but otherwise it was a good wheat year this year. So and also the investment in breeding has increased thanks to the Gates Foundation and other international donors. And also we are integrating the genomic selection quite heavily in addition to the phenotypic selection. I will talk about that in a few minutes. So perhaps what we see, the phenotypic data in the field, that is real, but there is a hidden part which we cannot able to phenotype. For example, we are based in Mexico, we may not get the reliable data from different partners. Then we can able to predict those data using our training set. So that helps us to make better selection for that particular trait. Yeah, so this is what the current application of genomic selection. So if you could see all these individual dots or individual elite line, which has been genotyped over the last 10-15 years. And this is our training set. And using this training set, we can able to predict all these traits. Yield related traits for irrigated, drought, heat kind of environments. And rust, wheat and rust, it is, inseparable. So we need to build rust resistance in our germplasm and also other diseases and quality traits and grain zinc. We are mainstreaming grain zinc means all our elite lines also have higher zinc in our breeding population. So basically using these models, we are stage 1 lines make this before we go for yield trials. In the pre-stage 1 stage, we make the predictions and select the lines, and in fact we kind of do negative selection. The low breeding value lines, we culling, we kind of discarding, and then high value lines, we advance them to yield trial because yield trials are quite expensive. So we need to prioritize what are the lines which goes to different target selection environments. So the phenotyping, it used to be manual in many days, many years. We go the bidders, we go with the paint, the spray paint, and we select. Nowadays we have other options like drones and all. We can able to make use of those data, images, because drones can fly within few minutes or few hours the whole station, and we can make use of those data. So that doesn't mean breeders don't have to walk, but still we can use make use of the additional data, the high throughput data from these images. So this is one of our recent publication from our quantitative genetics team that you can be able to improve the genomic predictions using the historical data. So whatever the dots which I have mentioned in that PCA graph, so that still we use as a training set. So that improves the prediction accuracy over the years for many traits. So this is the correlation between the observed values and the GBVs, or the estimated breeding values. You could see for the different yield traits, it varies from, the accuracy varies from 0.26 to 0.37. And for other traits like diseases is quite high and protein content is high. So perhaps we can use predictions to, discard the low protein lines, or high disease severity lines, for different environments. And also another improvement we have done is the rapid trait introgression through speed breeding. So whatever so far I mentioned about the population improvement of the breeding cycle, but on top of that, we do have this facility where we can able to grow 4 seasons per year. So basically seed to seed about 3 months. This is also established in Toluca. Here we use the special lights and optimizing the temperature. We can grow crop within 3 months. You can also see here using we use molecular markers, and then we make the backcross. So for example, if you want to bring these additional disease resistant traits or climate resilient traits for which we have molecular markers, so then we make the selection, and then we make 2 backcross or 3 different backcross, and then we derive lines. So this is typical marker stud breeding selection process. So this has been also running over the last 6 years at Summit, Mexico. So all these we do uh, to distribute to different partners, because whatever we do, the high impact research at CIMIT or in BISA, so that has to translate it as a varieties in farmers field. So that is why if you could see our seed multiplication in this northern part of Mexico, which is close to the US border, called state called Baja California. We produce all the international seed because that is a disease-free area, and we bring the seed to our main headquarters and then make the sets and distribute to different partners. And here you can see there's a portal where the partners, anybody can make the request, and then we evaluate if it is a genuine request. Then we make the shipment. The additional thing about BISA, so Karthik, how I'm doing with the time? I'm okay.
You
Uh,
you can proceed, sir, please. No problem.
okay.
You can.
Yeah. So since this is for India, we have excellent platform called BISA, Borlaug Institute for South Asia. I'm sure you may be here already. Part of this wheat, we send every year about stage one, like 5,000 stage one lines to our visa platform in Ludhiana, and then stage two lines distributed across 3 different sites between Ludhiana, Pusa, and Jabalpur. So those get phenotype, and then we get the data back to Mexico, and also these selections made in visa are also shared with other partners. So this is a field overview. So Karthik was placed based in Ludhiana for a few years. He knows the volume of work what we do in Punjab, and also we have other two stations. And one of the advantage of this platform is we can go for large numbers. So, for example, here we send more than 5,000 stage 1 lines for yellow rust phenotyping in Ludhiana, because the race differences between Mexico and also in South Asia. And by sending material early to Ludhiana, we can able to create the disease epidemic, and we can able to select yellow rust resistant lines. Similarly, for spot blotch, we do this in our PUSA platform in Bihar, Samastipur. So there also we inoculate, and we select for spot blotch resistance. So this is a huge progress across these three visa sites, and also other partners like IAWVR, ICR, and other state universities like Punjab Agriculture University, Haryana Agriculture. So we partner with all of them, and they take our elite lines, and then they evaluate and share the data. So thanks to all the partners, because we cannot do everything. So it is done in partnership with many partners in India as well. So every year also our visa stations, they have the field day, both the public and private sector partners invited, and they do make selections, and we do distribute seats to these partners. And the last thing to mention about the biological nitrification inhibition. So this is one of the translocation came from this grass they call Lemus racemos. So we got this translocation into chromosome 3bs. Basically, these translocations inhibits the nitrogen loss from nitrate to nitrous oxide, and nitrate. So from ammonia to nitrate and the nitrous oxide. So which means the you can keep nitrogen more in the soil near the plants for longer time, so that avoids the evaporation of nitrous oxide, that is a greenhouse gas. So that huge amount of nitrous oxide goes to the atmosphere every year, because thousands of millions of tons of urea or nitrogen is applied. So even if you could reduce 10% or 20% nitrogen, that's a big saving for farmers and also environmental friendly. So we have made huge progress also on these BNI lines. You can see now we have the BNI elite wheat lines, which has been tested across different locations. This is our field site in Mexico, Obregon, but also we have active breeding pipeline in Bisa for the BNI wheat. We are hoping the first set of BNI varieties will come to the market within few years. And also we are working on the funded from the Gates Foundation on the hybrid wheat, because what I mentioned, the pure line, the inbred program, we make 1 to 2% gain. That is still good, but we are expecting even hybrids can make more than 5-10%, 10 to 15% heterosis if you could exploit. So there is a new system called BLA, Blue Oleron system. It linked to the fertility gene. It's kind of patented trait, but still we have access to this system. So we are trying to make use of this system, and we are expecting this hybrid wheat also will be in the market by 2027, 28. And it is not, we are not forcing farmers to switch to hybrid wheat, but we will give opportunity if some farmers want to have hybrid over inbred, so they will have the choice to choose. So with that, I would like to thank you very much, and thanks to our program director Flavio for his active engagement. And if you have any doubt, I will be happy to take.
Thank you,
Yeah,
sir.
okay.
Thank you for the excellent presentation. We shall now open the floor for interaction and questions. So participants are requested to kindly either raise their hands or post their questions in the chat box.
Audience, do you have any questions? Sir, I remember maybe Professor Purchedu, please, you can ask your question.
No, no. Dr. Karthik, after you. Go ahead.
Okay. Sir, I remember in the varieties that with our team, with your leadership, we have developed around 45 to 45 ppm also in the stable lines. After that, can we get more, little more than 45 ppm around in high zinc lines? What is the situation now?
Yeah, the zinc lines are, it's also in pipeline. So, we cannot go very high levels of zinc because there is a trade-off with the yield. So we need to balance it.
That is true. Yield trade-off is also there.
Yes, if you for really high micronutrients, it is good, but you will at the cost of yield. So, farmers would like to have, more yield, more profit, and so these are
Yeah,
heavy trades.
however, the varieties released in South Asia are very useful to people.
Yeah.
At least we can overcome at some extent to malnutrition with the zinc
Yeah.
deficiency.
No, that is obviously we are working on zinc biofood. I did not highlight that one
Yeah.
much, but that's part of our
You can
breeding
please
program now.
tell about the varieties released in South Asia.
Yeah, if you could see a lot of these PBW 826 and HT, sorry, HT 3386, DBW 327. There's so many varieties of zinc biofortified already in the market. I think the estimate is about close to uh, 30 to 40% of wheat area in India is already with the slightly high zinc varieties.
High zinc varieties. Yeah.
Yeah.
Okay. With the
So,
CIMMYT and ICR collaborations, also you are working.
yeah, I said at state universities as well.
Yeah.
So,
Okay.
yeah.
Professor Purchedu, please ask your questions.
Yes, thank you. Thank you for your presentation. I have a few questions.
Yeah, please.
Years ago, CIMMYT planned to have 200 genotypes, considering that as a base of their activities, of its activities, consider them enough for those activities. Now, considering the progress in genetics and especially in genomics, do you still have this number as a guide or guidance, or you have changed things? That is the my first point.
Yeah.
The second point is we have a climate change. What changes, if any, on your strategy, breeding strategy? Is the plant ideotype still the same, or has that been changed?
Yeah, those are good questions. Actually, we don't have any limitation for the number of lines, or maybe in our crossing block we use 200 some numbers as target, because we cannot manage more than 200 parents. Maybe for the crossing purpose we use a few numbers, but obviously we don't have any restrictions on number of lines, because still semi-wheat program is like a numbers game, breeding program. Numbers game, we make thousands of head rows, and then we make selections. It's all about culling and making selections of good plants, which will become new elite lines. And the second question on the climate related, idiotypes. Yeah, so the thing is, especially in South Asia, the winter is getting shorter, especially in the northwest and the high protection environments of Punjab, Haryana, and other places. So we are looking for kind of slightly early maturity or mid maturity varieties, so that they can escape from the terminal heat stress. In addition to that, also we part of our routine selection process, all the elite lines, we expose them into different heat stress. We have selection environments in Mexico. We expose them into late zone trial, so that when the temperature is high, we can able to select the heat tolerant varieties. Similarly, also we do have the drought trials where we select the drought tolerant lines. We put very minimal water through drip pipes, only 120 millimeter water. So we will see how with this only 120 or 150 millimeter water, how the wheat can, how much wheat it can produce. So all these part of our testing process, and we are trying to maintain this maturity class and slowly moving towards mid early time.
Sir, what about Durham? Because Italy is concerned with the Durham and Dr.
Yeah,
Karim
we
is
have a
involved
Durham
in.
breeding program is strong as well. Unfortunately, not much Durham in India. So yeah, so we do have good Durham breeding program in Mexico.
For Italy, maybe here.
Yeah.
Audience, do you have any other questions? DK sir, do you would like to interact a bit?
May I ask, put a
Yes,
further
sir.
question?
Please, sir. Yeah.
Now, people is considering to have some delay in flowering or jointing and reduce and early ripening, so that reducing the period of
Grain filling period.
translocation of assimilates to the grain.
Yeah.
Now, any activity or any idea about the efficiency of translocation of assimilates?
Yeah, not much per se. But that is a good point actually, the grain filling period is getting shorter. So how efficiently the genotypes can translocate, the photo assimilates to the grain. So that's a bigger question. We need to see how we can able to phenotype that, from anthesis to grain filling. I mean, physiological maturity, how quickly, the grain filling period that. So I think we need to look at that point as well.
Our DDG, Dr. Nutan Kaushik ma'am joined. Ma'am, do you have any interactions?
Thank you. Thank you. It was a very good presentation. In fact, I just wanted to share regarding the malnutrition and zinc deficiency and so on. I would just like to share that at Amity, we have been working on zinc biofortification through nano zinc fertilizer, foliar spray of the nano zinc fertilizer. So what do you think? And we found that nano spray, foliar spray works very well as compared to the soil application. It's
Yeah,
more
definitely. Yes.
efficient, more good. So this is an instant. Any crop can be sprayed. So you don't
Yeah.
have to spend like years and years in breeding the crop. Rather than you just spray the zinc, nano zinc or nano fertilizer, nano micronutrients on the any crop, and then you get the fortified crop, fortified grains, and it get very well get translocated to grains. So
Yeah.
what is your opinion? Still, we should go for the breeding approach, or should we go only for the 4 years or go for the integrated approach? Like
Ma'am is
your
asking
because
breeding and
varieties, so
applied
many varieties.
sciences.
Yes, so many
Yeah.
varieties. Each variety is
Yeah.
different. The yield is different. Yield is
Yeah,
dependent
no, absolutely.
much of
No,
money is spent on the breeding and so on. So
yeah.
as a breeder, how do you feel about it?
Yeah, no, this is so in CIMMYT, breeding program is more of dynamic program. We don't do it just for sake of doing. So what you mentioned is that it is true. The foliar application works nicely, better than the soil application, and provided if farmers want to spray, that is
Yes.
one thing. And we have done some experiments as well in Mexico. The zinc sulfate, or it can be combined with these the other inputs like herbicides or the pesticide. When the farmer sprays the pesticide or fungicide, they can make some zinc, and it even have a good translocation into the grain in zinc. So I totally agree with you. So we don't have to spend a lot of resources for just breeding, but I think we should have this combined approach of the agronomic biofortification and the genetic biofortification.
We have done extensive trials I think we have done in more than 500 fields of this. This was supported by Bill and Melinda Gates Foundation, BMGF.
Yeah, no, that works nicely. Yeah, we have done also.
And it was wonderful results. And
Yeah.
so we are very much convinced that this approach works very well. and
Yeah.
can be adopted at larger scales. We'd like to
Yeah.
collaborate with you to get it the base for biofortification through the foliar application. So
No,
that
definitely. So please,
that could be
yeah,
one.
we can work together on that. Yes.
Okay. Thank you so much, Karthikeyan, for bringing Dr.
Thank you,
Velu
ma'am. Thank you so much.
Thank you.
Audience, do you have any questions?
Pleasure listening to your talk. It was a very wonderful talk. Yeah.
Yes.
Thanks, Dr.
Yeah, Dr. Valu, I was very impressed to see one point that you discussed over there, biological nitrification inhibition.
Yeah.
Very nice mechanistic approach you defined. We always talk about BNF, and whereon you are trying to stop the nitrous oxide gas emission in the environment, which is third in number after CO2 and methane. Sir, what kind of approaches, strategy you are deploying to denitrification step in the soil?
Yeah. So basically, this translocation in wheat, it promotes nitrifying bacteria near the rhizosphere, near the soil root zone. So that stimulates bacterial growth, and once nitrogen is applied, either we apply nitrogen, urea, or ammonia. So that bacterial activity helps to keep the denitrification. So that avoids
Actually, the
nitrate.
combination of rhizosphere is very important.
Yeah.
See,
No,
there's
these are some
molecular
exudates.
aspects, some enzymatic actions.
Yeah, some exudates. Some
Yeah,
exudate from the root zone
right,
promotes these microbial growth, especially nitrobacter and other things
right,
that
right,
stops
right.
the nitrification, no, and
Very, very,
denitrification.
very nice talk. Very nice biological nitrification inhibition step. That very impressive. That
Yeah, I
I'm
think
inclined.
no wonder you will see BNI wheat in the market in a couple of years. So
Right,
it's almost ready to be deployed to soon. Yeah,
certainly. Thank you, Dr.
in
Valu.
India,
Thank
in
you so
India
much.
first.
Thank you so much, sir.
Yeah.
Hopefully, we will establish MOU soon with CIMIT. So
Yeah.
we can collaborate, as ma'am said.
Yeah, especially for the BNI wheat, we have a special agreement. And if you are interested, then we can discuss about that. But there is no royalty such thing. But the translocation itself, there is some material transfer agreement has to be signed. Yeah.
Yes, definitely.
Okay, Garima.
Yes, sir. Thank you very much, sir, for your insightful and enriching presentation. So you may end your presentation now.
Thanks so much. It is
Thank
quite
you
late
so
for
much,
me.
sir. Really.
I will leave the meeting. So thanks so much.
For spending your time
Thank
in
you.
this time.
And my apologies for the hiccups.
No problem, sir. You have encountered the technical issues, and also it is midnight. We are grateful.
Yeah, it is midnight.
for
So
the
over
presentation.
12 a.m.
Yeah.
It's fine. It is a nice discussion. Thank you.
Thank you,
Bye-bye.
sir. Thank you so much.
It is now my pleasure to introduce our next distinguished speaker, Dr. Navdev Singh Bains. Dr. Bains is a distinguished wheat breeder, plant geneticist, and agricultural research leader, known for his major contributions to wheat improvement, molecular cytogenetics, and climate-resilient agriculture. He served as professor and director of research at Punjab Agriculture University, one of India's premier agricultural institutions, until 2021. He has also been a prominent faculty member in the Department of Plant Breeding and Genetics at PAU, contributing extensively to crop improvement research and academic leadership. Dr. Bains is internationally recognized for his expertise in wheat genetics, molecular breeding, disease resistance, and quality improvement in cereals. His research has significantly contributed to the development of improved wheat varieties with enhanced productivity, resilience, and adaptability to changing climatic conditions. Through his work in molecular cytogenetics and advanced breeding strategies, he has supported sustainable agricultural production systems and strengthened wheat breeding programs in India. An accomplished scientist with an extensive publication record, Dr. Bains has authored numerous high-impact research papers and has earned wide scientific recognition for his contributions to agricultural sciences. His research work has received substantial citations globally, reflecting the impact of his studies in plant breeding and crop genetics. During his tenure as Director of Research at Punjab Agricultural University, Dr. Benz played an important role in promoting agricultural innovation, strengthening industry-academia collaborations, and facilitating commercialization of agricultural technologies through various institutional partnerships and memorandums of understanding. He has also actively contributed to discussions and expert deliberations on climate change, sustainability, and resilience strategies for Punjab agriculture. Through his leadership, scientific excellence, and commitment to crop improvement, Dr. Navtej Singh Benz continues to contribute towards sustainable agriculture, food security, and climate-smart farming systems. With these words, I now invite Dr. Navtej Singh Benz to deliver his presentation. So please.
Sir, we welcome you. Thank you so much for the presentation. You have vast experience. I know I attended your several lectures. I think we hope to have your nice lecture today. Please start, sir.
Yeah, thanks, Garima, and thanks, Karthikeyan. So I'm just going to share my slides now.
Yes.
So I think that's now visible on your screens.
Yes, sir. Yes, it's visible.
Yes, sir.
So this, I'm going to talk about not a lot of information. You're already into your 5th day of the training program, the faculty development program on climate resilient agriculture. So I just kind of bring in a few thought-provoking kind of points for you to consider, particularly on the genetics and the genomics side. And of course, there's the information is a little is added there in the appendix. So I will just roll the slides in the appendix also, and then maybe they can be useful if you kind of go to them later on. Right? So this is, incidentally, this is a painting from Van Gogh. And it's a disturbed wheat field. And Van Gogh, the very celebrated artist, he was under some kind of a mental disturbance, progressive, and his paintings got more and more disarrayed and disoriented and with these kind of strokes, and then kind of prized for that actually. And today our wheat fields are getting disturbed more from external reasons. And you know, all the issues are related to climate change. And so this is a very ominous picture. And today, incidentally, is also the World Environment Day. And so these are the main strategies. So this is, what could you do to address climate change crisis in agriculture? Either you go for climate smart crop varieties or you go for climate smart agriculture practices. Often this is a kind of a division is made that this is an adaptation strategy and this is a mitigation strategy. And we are primarily, my topic would be on the climate smart crop varieties. But before I go to that, let's also consider that agriculture is not just the victim of climate change. It is a perpetrator as well. And I have had some research on this in collaboration with my agrometeorological colleagues at the university. And we found out, we had a big data set around 50 years. And in Punjab, to start with, rice was not spread all over the state. The central districts were growing rice, and the adjoining flanking districts in the north and the south were growing other crops. And what this agromet data told us
50 years, 1970 to 2020, that there is a clear, distinct, and very significant and with a very high regression value change in night temperature, minimum daily temperature, in the rice intensive districts. 0.054 degrees C per year. And you can imagine the kind of change that would have happened over these decades. While in the flanking regions, the changes were non-significant. And there was a lot of corroborative evidence in the form of changes in humidity, changes in sunshine hours, and all those things kind of corroborating that the climate change has accelerated. And the reason was simple. In these areas, we get actually a shallow ocean of water, because this is more than 3 million hectares and contiguous. No other formations in the soil, all flat level topography. And what we are getting is standing water, and it changes everything. It changes the entire response of the land and impacts the climate change severely. And though it is very hard to make a causative connection there, but we find that once this area under ice exceeded 2 million hectares, our monsoon rains have plummeted. Last 20 years, and now it's all more than that, it's 25, 26 years, the monsoons have been deficient by 20 to 40% year on year, except for just a few exceptions over these years. So this is the kind of thing. Agriculture is a kind of a dual issue there. And you also probably most of you have seen this, that these are the NASA GRACE satellites, and they check for gravity. And the gravity underneath this area where we are sitting, including your area, the northwestern plains of India, the gravitational pull has reduced as a result of water loss from the underground reservoir, from the aquifer. See changes of such magnanimity, such huge changes. So this is the kind of precarious situation. There could be so many other points to make in this regard. But let's shifting to the genetics part a little. This is one of those projects when these things came up first into our kind of urgent agenda. Some 20 years back, I'm reminded of this project we took up under the DBT. And here we were trying to use land races. It's good to follow Velu, a very renowned wheat breeder. And here I'm talking about wheat again. And here the donors for drought and heat tolerance, we were looking for these traits in the land races. The tall varieties, the varieties that were being grown before Velu's Institute Summit brought in the semi-dwarf varieties which solved our hunger problem and food security problem. But before that, we were growing these C306, 273, now the C varieties in this area, all of North India. And these were without irrigation, without fertilizer, and of course, very tolerant to heat. Could we use these to kind of get something back into our improved varieties? So we did a joint project with our biotechnology and the plant physiology divisions. And we found out something that on the right hand of the screen, you could see that all the tolerance traits that came from these tall varieties were tied up with their tall stature. They were storing more water-soluble carbohydrates in the peduncles of these tall plants. And their chlorophyll was low. For that reason, the productivity was low, but for the same reason, they were trapping less of solar energy and experiencing less of terminal heat stress. Now, to our great dismay, we understood that these tolerance factors are not transferable to the high-yielding dwarf varieties. So that's the question. That's the thought I want to provoke in the faculty members attending this program. So, do we have the basic materials and informations to develop these climate resilient crops? Or we are just doing some cherry picking here and there? But even a more serious question, do we know what we need to do in this case? And I'm talking in the context of genetics, genomics, the biotechnological revolution. So, Miss, this is I've been as a breeder. I see that these different crops. Means it's now something due to these new technologies that we can look across the crops. Means otherwise, as a wheat breeder, I was restricted just to be talking, thinking, deliberating, working with wheat. But these technologies now they make it possible for us to look across crops, because we can look at DNA across these crops. We can look at transfers across these crops. So wheat is a cool and aerobic adapted crop. Maize is warm and aerobic. Rice is warm and anaerobic. So our design of future crops, say a wheat which is more tolerant to the warmer conditions, a maize which is more tolerant to the flooding conditions and all that. That's what the climate variability is doing to our crops, our cropping systems. So it boils down to sourcing these adaptations from across the crops. Are we in a position to do that? So that's the question. It looks very attractive. And I guess, though it may not be formulated in these kind of words, most of our advanced genetic manipulations are working towards this. But do we know what is the basis of these adaptations? How many genes are involved? Whether these are the metabolic factors or master switches, or what kind of, where do they lie in the hierarchy? And another question is, because we have seen that as we have moved toward higher and higher productivity, we have inadvertently sometimes opted for narrower and narrower adaptation, which means we provide them the best management and the least shock to higher productivity. So, is a narrow adaptation a cost we pay for higher productivity? Or question, I'll flip the question. Is it possible to have a resilient crop with high productivity? I'm sure these questions are going to be center stage, and people at your stage of your research careers, you are going to encounter and address them and hopefully solve them. So this is the across diverse species, across diverse adaptations. Do the differences that we are looking at now allow us to design new genotypes and extend the adaptation regimes? So much of this, I'm going to transport you into a different topic for a while. Much of this answer to this question, besides the technological part, besides the advances we are doing in all the genomics research, there is an underlying fundamental question which needs to be answered, and which people like you doing advanced research must also consider, which means that do we understand the genotype-phenotype connection? Because our ability to work, modify, reorient, change the genotype is immense. And we hope that's going to give us the desired phenotype. How good is that hope? So I have it's something for you to think about. We can't go into much detail of this, but probably people in working on the genetic manipulation side, genetic engineering side, must understand that a lot of genetic variation is adaptively neutral. And this has been known to us for decades. And often I feel that this is not being properly considered in our learning curricula, the textbooks, and other places. Most genetic variation that we see, most of the DNA, the huge polymorphisms that we see, have nothing to do with adaptation, or nothing to do with the current adaptation, I might say. So that's a big deal. That's a big deal. And you need to see how and why of this and how to get over it. That's the issue. And then there are other points to be considered in this genotype-phenotype connection. The developmental switches, some of the master genes. Once they change, all the genes, or many of the genes downstream, their relationship to the phenotype changes. So means that this is when you're deep designing a new crop for climate resilience, this is something that you have to consider, or even better, that you might deploy, you might use to get the results you are looking for. And of course, I'm trying to here explain that how these things happen, how a major change in a developmental switch often renders a plant handicapped. But out of that handicap, a new evolutionary pathway can arise, trajectory can arise. We have examples in evolution. We have the cabbage and the cauliflower. This is a totally different plant type compared to the ancestral brassica, the sarson crop that you grow for oil. So this change happened, it's a handicap, but then it leads to a new crop. In case of maize, that's another interesting example. The maize progenitor is so unlike maize. The plant type, the grain type, the kind of cobs it has, it's totally unlike maize. And then there is a single or a few major developmental mutations like PB1, which leads to another totally different. In fact, till this happened, the New World, the Americas, did not have a cereal crop. And for that reason, they were left behind the other world civilizations. And when Columbus reached, they were pretty still somewhere kind of lost in their civilizational program. And then there's another, maybe this is the final issue with this. The enzymatic machinery, because we are thinking of combining the high productivity with better adaptation to some conditions and maybe changing adaptation to changing conditions. But we find much of the enzymatic machinery has a very narrow bandwidth of adaptation. It may not be possible to combine these traits with each other because they will put constraints. If it is adapted to one kind of temperature regime, that plant will not do well in the changing regimes. So back to the question of adaptation versus wider adaptation versus productivity. So these are all the things we need to consider. And of course, beyond that, we need to have the ability for multiple coordinated genetic changes. And there is such a, I say, flurry of activity on that side. Every day, new technologies coming up. CRISPR has opened gates for just targeting anything and changing it. And the technology spectrum is advancing. And of course, the new means when we're looking across the genotypes, we are looking to have information or understanding of genomes across species. And people have put in concepts like superpan genomes, very high fidelity, very precise genotyping and sequence, high platinum quality DNA sequence across the species so that we can track down minor differences which might be contributing to the adaptation differences. And there is this concept of intermediate phenotypes that instead of looking at the final adaptation traits like yield or performance and such things, look at some of the steps at the protein level, at the transcription level, at the metabolite level, and then use these intermediate phenotypes to assemble your target, I would say, idiotype or plant type. And of course, the phenomics area is going to be so important in this because the precise environmental conditions in which you can really see the response. Because nature doesn't afford you this opportunity often, and this has to be done under controlled conditions. And then because the data at all omics layers is growing so fast, even within an omic layer, it's becoming hard to integrate. And now we are like looking to integrate it in the multi-omics across the layers till we reach from DNA to the phenotype. So this is a tall order, I would say. Unless we are able to have a thread there, unless we are able to have a connection across these omic layers, I don't think the genomics assisted breeding is going to be as powerful as we imagine it could be, because only when we tie up this sequence, that's going to happen. And of course, there are already a few things on the ground. You have been told about this. It's been the news is there. India has world's first genome-edited rice varieties, which have been announced. But then that brings you to another aspect of these technologies. These are still some distance away from actual deployment in the farmers' fields because of IPR issues. Even these ministries and the ICAR, very well endowed bodies, even they are facing IPR hindrances. Hopefully, they get them cleared quickly, and also for at the institutional level, and some of that gets channelized to places like Amity and other universities. So this is, but just to tell you that it's not all talk now. So we are still not there, but the work is in progress. So thanks for these, just kind of throwing around some thoughts. But I've also added some slides if somebody would like to go to them later. People are now giving out these genomic assisted breeding programs or the flow diagrams for this, and the flow diagrams for integrated genomic selection. The papers are now coming out routinely. And of course, the super pangenome. I suggest those people who are in this area of work must go to how the super pangenomes, the platinum standard reference genome, how they're going to make a difference when we're looking at adaptation across the species. And of course, they have already tried to look at some of these species-specific variations and how, or even within the species groups, and how they contribute to adaptation. Some examples are already available to us. And intermediate phenotypes I mentioned. So this is also an area of work. Those who are on the software side of this work, they can really make a huge contribution to this opening area of research. And the integration of the multi-omic layers, that's a big deal. And though I do not know, all these softwares are evolving now by the day and by the night, and you have so many programs coming up, but this has to go hand in hand. As we make progress on the conceptual side, on the field side, lab side, and this software side has to keep pace. So this is a highly networking and nested program. And of course, people are already, though it's right now they're doing it only in the model crops or the model species, the genotype phenotype maps. And it's only now being done theoretically, but I guess the GP maps are going to be there for crops. And that's what we have been talking about. That's what we are looking for. So different issues, the kind of conceptual issues that I mentioned, also being taken care of in this GP maps. And of course, the CRISPR, I need not develop on this. So many people talk about it. And this has become so now pervasive and so important. So thanks again. So this is a scene from Van Gogh when he was in better shape. Hope we too, our world, let's wish on this Environment Day, also gets into this kind of better shape. So thanks for this very patient hearing. And any discussion points or questions.
Thank you so much, sir.
I'll be happy to take that.
Yeah, I think.
Thank you, sir, for the excellent presentation. So we shall now open the floor for interaction and questions. So participants, they are requested to kindly either they raise their hands or post their questions in the chat box, please.
Audience, do you have any questions?
Hello.
Yeah.
Yes, sir.
Yeah, Professor Purchedu.
Good afternoon, Dr. Bas. Baljinder.
Yeah, Baljinder, yes.
Just wanted to say hi and thanks for the wonderful lecture, sir.
Yeah, good for recognizing me without my turban today. I'm at home.
It was really nice to hear you, sir. Yeah. Thank you, sir.
Professor Purchedu, do you have a question?
Well, I want to warmly thanks the speaker because he has expressed all the thoughts I have in mind and put in so brilliant way what I am not able to express even on my own language. And so thank you very much, because that is really points that scientists working on wheat or rice, on breeding, on genetics and breeding, on crops has in mind. And unfortunately, not all the points have a clear answer. But thank you for putting on the table.
Thanks, Dr. Purchedu. Thanks so much.
Thank you, sir, for your contribution to science is vast. In Punjab, the situation of wheat production, what is the situation, sir? Can you describe now about the wheat production?
Yeah, well, this time again, we are suffering some productivity losses.
Yes.
Some 2 quintals on an average per acre due to the not typically terminal heat.
Yeah.
Due to a lot of climate disturbance. So we were having volatility all through the harvesting season. And it's very hard to put down your finger on which climatic factor really reduced the yield levels this time. But almost across the state, and I guess across Haryana and Western UP as well, the wheat productivity has suffered. And there is a clear climate angle to that. Though
Yes.
the climatic factors were a little more complex, I would say,
Yeah.
and multiple. So we
you
were
are saying
having
apart
hails
from
and we were having
genetics and all,
thunderstorms.
we have to focus more on climate resilience, more on increasing trees to prevent the pollution, lot of things also.
Yeah, no, this is an issue which now then a whole generation is actually faced with this crisis. And Dr. Enrico and myself in the older generation, I think now we need to be on this side of the work, not just on the productivity side. We need to be, we are really in a kind of a very tough situation.
Yes.
And I'm really happy that your university has taken a call in terms of this development program to talk about this.
Yes. Thank you so much, sir. Yeah. So apart from major cereals, Punjab, they are concerned on minor cereals also, because I know in past years also rice production also they concerned on rice production. So what about millets and others in Punjab situation?
When I talk of Punjab, actually, means see how the things become systemic and kind of entangled.
Yeah.
We can do a lot of talk about the millets, but then we have designed or kind of moved to production conditions, irrigation and fertilizer, and the economic expectation of the farmers. So
also, I remember during
we are in a catch-22 situation with respect to millets.
Yeah.
From the typical millet areas, you don't have much of marketable surplus.
Yes.
From areas like Punjab, Haryana, and Western UP, where everything comes to the market, the conditions are not very conducive to the growing of millet varieties that we have presently.
Yeah.
So as a breeder, I'm looking at this from this perspective that we need to have millets, which so it means millets has undergone very little breeding work for this kind of a target.
Yeah. During our past conversation, you also said, when I was in Ludhiana, about durum wheat. During ancient time, Punjab also producing more durum wheat. I remember you said me, but now the situation is changed only for bread wheat. So in future, whether in Punjab Agricultural University and other universities, they also concerned with the durum wheat production, sir.
Yeah. Now the production systems are actually evolving in a direction where the food security is, of course, important. There was a lot of concern when the productivity dipped a little during these years for rice as well as wheat.
Yes.
But I think the consumers are now going to lead
because
the diversification impetus.
Major
whether
crop
it is for
some people having this gluten allergy, some
yeah. So means they and of course the processing pipelines are going to be important. and the people are taking an interest in Sapphirococum. There is means that they have sent out means people outside the academic area have kind of popularized varieties of Sapphirococum. And Sapphirococum is the original Punjab wheat, the old Punjab map. If you see, they are the even prior to our so-called Desi wheats, they were there. And probably the scientific fraternity is now waking up to these opportunities, actually.
Also, we were talking about the tritorium.
Yeah, there's so many
things like,
possibilities, actually. Yes, yes, Karthik.
Barley grass. So
yeah. So there are many opportunities.
Many things, yeah, opportunities. Okay, sir. Thank you. Audience, any questions from audience side? No. If not, we can conclude the session. I think Dr. Garima, we can.
Yes, sir. Thank you very much, sir, for your insightful and enriching presentation. So you may leave the meeting now. So
thank you. Thank you very much.
Thank you, sir.
Thank you, sir, so much for providing a valuable lecture. Thank you, sir.
So with this, we conclude the session 1 of day 5. And I sincerely thank all our esteemed speakers for sharing their valuable knowledge and experiences, and extend my sincere gratitude for taking the time to share your expertise and experiences with us. We truly appreciate your valuable contribution to this faculty development program. I also thank all participants for their active involvement and enthusiastic participation throughout this session. Thank you all for your presence and engagement. I would like to inform all the participants that the final feedback and final assessment of this faculty development program will be conducted from 2 p.m. to 3 p.m. today. So I kindly request everyone to be present and participate in the assessment, as it is a prerequisite for the issuance of the e-certificates. We look forward to your active participation. Now it's time for lunch break. I request you all to join at 2 p.m. Thank you very much.
Deepika, may you please proceed, paste the link 223, provide 50 minutes time to perform the test, and then henceforth we shall discuss thereupon.
Okay, sir. Good afternoon to all the respected resource persons, esteemed faculty members, and dear participants. Over the past few days, this FDP has served as a vibrant platform for knowledge sharing, meaningful discussions, and capacity building in our academic journey. So, to proceed with this afternoon session, we firstly begin with the quiz-based assessment of participants, along with the feedback from the participants, which is conducted to evaluate the learning outcomes of this FDP program. The link for both feedback as well as your quiz-based assessment will be shared in the chat box. You are kindly requested to fill both the quiz-based assessment as well as your feedback form, and the time given will be up to 2:50, as after that at 3 p.m. we'll start with our valedictory session. So the link will be pasted in the chat box. The link has been shared in the chat box. Every participant is requested to kindly go through the links and fill the feedbacks as well as your quiz-based assessments.
Okay, we shall meet at 2:45 then.
Yes.
You may proceed further.
Okay, sir. So now we are going to move forward with our next important segment. So in this segment, I would request few participants to kindly share their experiences and key takeaways from this FDP, because it not only helps us to reflect on our efforts, but also guides us in improving the future initiatives. So I request any participants to kindly share their experiences about this FDP.
Any one of you, please share your thoughts. What did you suggest us? And what did you learn? Learning outcome, please.
Participants, you can please share your experiences, what you learned.
Sir, can I come in, sir?
Why not? Why not?
Yeah.
Please,
Yeah.
Dr. Singh.
Thank you, sir. Thank you, Dr. Chaudhary. And
Yeah.
thank you all the eminent organizer of this course. So it was lovely and very useful, rather for people like me who are in the development sector. So sometimes we get involved so much in the development sector that we, have certain gaps in terms of the scientific knowledge and scientific developments that is happening across the world. So it was really a wonderful opportunity and learning experience to listen to all the participants. And especially I like the session by Dr. Navtej Vance. He has been guide for us also. And from Tata Trust, we also had one grant to PAU where we worked on the accelerated breeding program for wheat. So for me personally, sir, it was wonderful experience to, learn and share. Thank you, sir.
Thank you, Dr. Singh. We are delighted to have you with us today. Dr. Singh, I think I reported you are for last few days with us from first day.
Yes, sir. Yes, sir.
Yeah, I have seen you many times. Thank you, sir. Any other participants, scientists, research scholars? they may share their views over here. Dr. Karthik, you may invite Professor Enrico as well if
Professor Enrico Porcedo. Professor, are you in online? I think he's in online.
Yes. No.
Professor, please give your feedback and also your support guidance for us, as you have enriched with a lot of experiences and knowledge.
I didn't expect to have to to report about your exercise. It was a brainstorming, very excited at certain moments. Others were just other presentations were just a report of what is happening in India or other parts of the world. Some enlightening points were very well presented, very stimulating, and others less. But that is, I think, that is normal in such kind of exercise. You see, you have opened the discussion to a larger presentations, scientists from larger parts of the world, not restricted just to India. And I think we all gained for such information, for so large information. As I mentioned earlier, some presentations were very stimulating. And in terms of knowledge, in terms of ideas for future. I am curious to ask the organizers, how do they, not how do they judge the presentation and the discussions. I'm going to ask them where they benefit for your plans. I understand that university is going to restructure courses, I suppose. And so that is, were those presentations useful or not? Also, I don't know exactly what is the level. I understand that you have a number of universities with different importance, with different levels of education. And I don't know exactly where you want to put yourself, at what level. So I'm curious to learn from you, from the organizers. Were those presentations useful? They were very diverse, as I mentioned. I did a presentation for agriculture as such. Others were more on specific pieces of research. Just a ordinary scientific meeting, a presentation, a piece of research in an ordinary meeting, annual meeting or so. So I'm curious to learn from you the usefulness of what was presented and was discussed. Not knowing exactly what is the position of the MIT university in India.
Right, Dr. Enrico, we are very grateful to you, sir. And you provided a chance to speak before you as well. With respectfully, the workshop was organized to keep things in the mind that what nowadays we can improve in agriculture under adverse conditions, you know that. So climate resilient agriculture, or so-called climate smart agriculture, what techniques, what approaches nowadays people's or the researchers all around globes are going to deploy, and what will be the implications these techniques shall be incorporated in our academic programs, not only for the graduate levels, but for the postgraduate levels, and what kind of techniques will be worthy for the farmers as well in terms of the cost effectiveness. So the things were in my mind, my team also, my director also. So we moved ahead to put all these together, not only for the academics and the science, but we were looking towards the transcend approach, lab to land work. A socially inclusive framework was decided in our mind also. We all sat together to analyze the things. What do we can do? What kind of the disseminations shall happens globally by putting such efforts? So we moved ahead. We invited you all great personalities, all scholars working in the different areas relate with the soil to productivity. The soil microorganisms, microflora, rhizobium, rhizosphere, and then what implications will be incurred upon deployment of these microorganisms towards the plant and crop sustainability. So such kind of things, but we were not restricted to the deployment of that approaches. Rather, we were deciding to deploy the gene editing approach that many lectures we have seen over here, RNAi, CRISPR, and genome engineering as well. So phenotype, genotype, relations, and certainly the how we are going to deploy these approaches to enhance the soil productivity and crop productivity in toto, holistically. So now we are going to make a report, a kind of the policy making decisions from the university level. The higher officials will sit together. A compendium shall be designed and we will try to publish it somewhere with the good publishers. That was the outcomes in terms of the policies. So this is a gist for this workshop, sir. Thank you.
May I ask a point?
Yeah.
You are preparing people, two classes. I feel that agriculture needs, or the agricultural system needs two types of scientists or two types of technicians. Those who go to explore science and bring new ideas and bring new knowledge and new ideas and new technologies, and those who are on the field and in contact with the farmers.
Right.
Now, on the first case, you may plan to organize teams that interact scientists on different fields, on
Right.
restricted fields of activity of research, and they needed to interact each other on their own field.
Right.
On the other side, you have people interacting with the farmers.
Right, sir.
And farmers more and more will ask why,
Right.
not how. I grew up on a farm family,
Right.
and my brothers are still, two brothers are still working on agriculture.
Right.
When I go home and have a contact with them, we are discussing about agriculture, they always ask me why, not how to do this, how to apply, but why. Why that happened? We have a field with a plots where there is no vegetation. Why?
Oh.
So this makes your job more difficult than
Right,
expected.
certainly. We shall move ahead by deploying this approach, how and why. So that's why we organize this, and we shall deliberate the same in the near future as well. Thank you, sir.
Thank you.
May I, Dr. Enrico, answer few of your queries?
Pardon me?
Dr. Enrico, as a director of this institute, of course, we are mandated to carry out faculty development programs so that our faculty gets updated knowledge, of course, newer knowledge, and acquire them to practice them. First, during education of our students. Second, to write projects, sometimes to collaborate, to network. And then we have centers and we call the farmers here, or we go to their field and we conduct outreach programs, farmers outreach program, where we answer each other's why and how. So that I'm sure we are not doing the work, like 100% we are able to satisfy. There are plenty problems with Indian farmers, or we have plenty problems which we have to understand from their perspective. Very simple example is the rice wheat cropping system. Right. Our means wherever this has been adopted, the water abstraction of the groundwater is immense. Yet we are unable to. A government policy shift has been done. Scientists have worked towards bringing some early variety of rice so that you can use, or sometimes direct sowing so that water abstraction is less and all that. But yet we are unable to convince our farmers to adopt. So it's continuous process and we keep doing it. And of course, I can only talk about my institute, where we have MOUs with some international, of course, they are nationally placed also, organic organizations. Now, they work on the farm level with the farmers, execute these things. Since ours is organic agriculture, so we promote organic agriculture. So these are continuous issues which like we have to address, and we have our extension department and center also. It is not easy job to convince farmers, because again, why? And then of course you have to show whether this is financially profitable sorry, for the farmers, because they are taking the rice wheat cropping pattern because of their financial gain. They very well know that they are ruining their soil, or water abstraction from the groundwater is happening gradually, or maybe many places have degraded soil now. But even then they do it. There are complexity of issues which needs to be addressed, and it's not so easy. Agriculture is very, very complex system in our country, especially where farmers are very resistant.
Everywhere. That was my first sentence. The first day, agriculture is complex.
Yes.
Thank you for the opportunity to participate
Yes,
on
yes.
this meeting.
Thanks
I'm very grateful for that.
thanks to you that we could hear diverse ideas and opinions and facts, of course, scientific facts. That enriches our knowledge, and we also start exploring whether we can implement these such things here, right? If the conditions are similar, or can this be adopted, all those questions starts in our mind, and that's what is important, to trigger the thought. So FDPs are the best ground to trigger such thoughts, right? Scientific thoughts.
Thank you.
Yeah. Over to you, Deepika. If anyone else wants to share their feedback.
If anyone wants to share their feedback, they can please. Otherwise, we'll move towards with the valedictory session.
Check if they have written something over the chat box or anything.
No,
You're
ma'am.
free to write in your chat box if you're if you have something to share.
All right. Thank you, ma'am, and thank you, sir, for your thoughtful reflections and valuable suggestions. So, as we now move ahead, it is my privilege to welcome you all once again in the valedictory session of this faculty development program on climate resilient agriculture and allied technologies for net zero and socially inclusive framework. So now, firstly, I would like to invite the co-convener of this faculty development program, Dr. Karthikeyan Tyagarajan sir, to kindly present the outcome of this FDP, highlighting the key achievements and impact of this program. So please. So your mic is mute. Can you please turn it on?
Thank you so much, ma'am. I'm Karthikeyan Tyagarajan, Dr. Karthikeyan Tyagarajan, working as an associate professor in Amity Institute of Organic Agriculture. I'm also a co-convener of this DFI program. So it's my privilege to provide some sort of a report as a presentation. So the report on faculty development program on climate resilient agriculture, allied technologies for net zero and socially inclusive framework. It has been a wonderful program, and we have covered multiple aspects from different faculties globally and also from India. The faculty development program on climate resilient agriculture and allied technology for net zero and socially inclusive framework was organized by the Amity Institute of Organic Agriculture under the aegis of Agriculture and Allied Sciences domain, Amity University, Noida. The 5-day online program consists of 30 hours aimed to address the urgent challenges posed by climate change and explore sustainable agricultural solution aligned with national and global levels. So climate change driven by greenhouse emissions, temperature fluctuation, and environmental degradation significantly impact agricultural productivity and food security. This FDP focused on equipping participants with knowledge and tools to promote climate-resilient agriculture, sustainable and inclusive agricultural system in line with the vision of our Viksit Bharat 2047. It has objectives. I'm just reading some of the objectives to enhance understanding of climate resilient and sustainable agriculture, to integrate modern technologies such as artificial intelligence, Internet of Things, and data analysis, of course, using artificial intelligence to promote net zero emissions strategies in farming systems, to strengthen research innovations and teaching capacities to support inclusive agricultural development through policy and community-based approaches. So for this lecture, we have 5 days of seminars, presentation from prominent Indian speakers and also from abroad. And it has 5 days with 5 themes. The first day, the theme, it is entitled as Climate Resilient Agriculture, Concepts, Frameworks, and Global Perspectives. And of course, Dr. Professor Enrico Porcedo was a senior professor. That's why we invited him. And he was presenting about the situation, food security situation in India, how to overcome some kind of problem using genetic and agronomical inputs. That kind of things Dr. Professor Porcedo described well, and also about the nutritional aspect, how to improve, apart from calorific requirement, how we can improve the nutritional aspect, and what kind of other sources he also described very well. So we are grateful to sir. And then Professor Brinchish Sarma presented about the plant microbe synergy for climate resilient crop protection. So he was taking a crop, specifically chickpea, and their interaction with the Trichoderma asperellum species, and how it is increasing phosphate solubilization. Apart from that, also he was talking about salinity resistance. So by incorporating genetic things, he also provided microbiologically how we can make a biopesticide, phosphate solubilization, and then about the gene, like especially about, he was talking about basic helix specific genes for nitrogen use efficiency as well. Then subsequently, Dr. Mallikarjuna Swamy, he was a former scientist from IRRI, currently working in a tissue culture laboratory as a director in Philippines. He was talking about biofortification strategies, because since he had plenty of experience of more than 15-20 years, he's talked about that with the title of progress in breeding for healthier rice varieties through sustainable agriculture. So he was talking specifically on how he was contributing for the zinc yield along with the teams of IRRI, and also he was telling about vitamin A, golden rice, how on the genetic basis, he was focusing specifically on the genetic basis to improve the zinc biofortification. Eventually, Dr. Patricia Galefi, former ENIA scientist and currently associated with the University of Rome. So Dr. Patricia was talking about specific things in climate, agriculture, and the future of agriculture. So she was talking specifically, I remember, about the zero tillage practices. So zero tillage practices is really we have to take into consideration, because so that we can avoid the emissions of greenhouse gas. So she was also talking about artificial intelligence and IoT, how we can incorporate these things to reduce the emissions. And also she was expressing about her previous, I was also involving with the DR1 gene, how it is contributing DREB kind of gene, dehydration responsive element binding factor, dehydration responsive factor with alternative splicing, how they are also playing a role to mitigate this kind of drought and other stress. So that was the thing. During the second day, there were 5 speakers based on the theme of climate-informed soil, crop, water, and post-harvest management in climate-smart agriculture. So initially, the session was started with H.B. Singh sir, and he was talking with the topic of management of soil-borne diseases using microbial pesticide. Nowadays, chemical pesticides having big problem. It is a persistent in soil for long time. It will cause multiple problem. Even it can act as a carcinogen, and also once we should consider with this kind of program, we should have urgency for the use of microbial biopesticide. That was he was insisting to use Trichoderma hasniarum, and then Pseudomonas fluorescens, excellent biocontrol agents, and also Bacillus subtilis, and various microbes and endogenous microbes, endogenous microbes. Also, he was talking about Verticillum lecani, and then Vivaria baviana. So a lot of microbes. It was so much interesting to know about these kind of microbes, and it will give a lot of awareness among the scientists and young students as well. The Siddharth Tiwari sir was eventually talking about the precision genome editing. It was a wonderful technique, because CRISPR-Cas9 is playing a major role nowadays to save the time. If you go through the QTL mapping, GWAS, whatever study, it may consume some time. So we can, if we go through this biotechnological aspect, especially CRISPR-Cas genome, through metabolic engineering, and how we can increase yield as well as nutritional, apart from calorific value. He was focusing specifically specific nutrition in terms of provitamin A and iron, ascorbic acid, vitamin C. That was where the compounds or the biomolecules he was focusing on, and he was very successful. and got an award from Dr. APJ Abdul Kalam. And also he was clearly describing how through gene knockout or gene editing, or we can knock out or we can also edit using prime editing aspect. And also he was presenting such a nice lecture. And subsequently, Dr. Debabantya Mahapatra from ICR was talking about post-harvest engineering, grain value addition, and smart storage technologies. Yeah, after harvesting in India, sometimes you have to think. As I stayed in abroad, I could also see the difference. Post-harvest loss in India is really more compared to European and all, because we need to also overcome this kind of problem. So particularly Dr. Mohapatra, a kind of scientist, they are working on this to overcome rancidity. Because if the grain is not properly processed, it may be having oxidations, and due to the presence of the lipid molecule that causes rancidity and foul smell, and also it's not good for the health. So how to overcome that thing? So we can also increase shelf life. And on the other hand, Dr. Mohapatra also presented anti-nutritional factor. For example, when we are consuming any kind of nuts, it has phytic acid. Sometimes we consume like almond directly, it is phytic acid. We should have that awareness. So how to remove the phytic acid? Simply by putting imbibition. Soaking in the water, so you can put some salt, you can overcome that. But they are doing systematic practices with ICR to overcome tannins, phytic acid, oxalates, kind of anti-nutritional factors. So subsequently, Professor Arun Kumar Joshi, sir, he was a former BISA director. And his lecture was also very inspiring. He was talking about how we can do increase productivity with early sowing, especially using heat-tolerant lines. We can do early sowing so that we develop in CIMMIT with Dr. Joshi and all. Some kind of lines. If we have early sowing, we can increase the line, because early sowing means you can harvest her early before terminal heat. In that case, it will not suffer to the terminal heat. Although they are also developing heat stress tolerant lines, but still we need to think of early sowing kind of strategies, and then to get the economic benefit. And he was talking about biofortification. He compared globally the situation of a farmer, what is the income. When he was talking about it, was really, we need to think of that. Sometimes some farmers are getting very low income. They have to increase because of this drought strategy, overcoming strategies. Then, so subsequently, Professor Bhupender Giri from University of Delhi was talking about shielding plants from environmental stress, the power of microbial inoculants. See, most of the speakers, they are really concerned on natural way of mitigating biotic or abiotic stress by using the existing natural microbes, resources. Dr. Giri was giving a excellent talk of symbiotic, pre-symbiotic, and post-symbiotic relationship, and how this strigolactose playing a role to establish the specific compound strigolactose playing a role to establish the relationship between the microbe, that means the arbuscular mycorrhizae, and the plant vesicular. Previously, it was called as vesicular arbuscular mycorrhizae and plants. And he also suggested specific genes, E node and symbiosis receptor kinase, calmodulin modeling kind of things, and also said specific upstream gene, salt overly sensitive, SOS gene, that HKT, high potassium, these genes are really taking a major part against salinity tolerance, which is really threat to the world. That kind of strategies is really we need to appreciate from Dr. Giri and eminent scientists. The fourth day, there were 4 speakers were talking on the theme entitled precision agriculture technologies and AI tool usage in climate smart agriculture. So in this session, we have also included the eminent scientists who are having enriched knowledge in microRNA. MicroRNA is small interference RNA. For instance, Dr. Niti Sanan Mishra is eminent group leader and scientist from International Center for Genetic Engineering and Biotechnology, was talking in the title of rewiring stress responses in crops, microRNA as drivers of climate resilience. So we need to consider, apart from protein and all, if we go in depth, microRNA, small interference RNA, how they are playing major role. In fact, Dr. Mishra also created artificial microRNA. Generally, plant produce their own microRNA, but they have also tested if they could produce artificially and how they can do, but by the injection or introduction of this artificial mRNA to upregulate this important gene and downregulate the unwanted genes. That was a wonderful lecture about this microRNA and siRNA. Then eventually Dr. Akash Shah from our Amity University, Amity Institute of Information Technology, was giving a good idea about even layman can understand about the data. If somebody, they don't know about scripting, because we need scripts, Python or language kind of scripts, we can also learn stuff, but he also provided data user-friendly database, how we can do artificial intelligence for agriculture, how we can do with data analysis using specific software, incorporating Internet of Things, AI technology to make drought resistant analysis as any kind of climatic resistant related analysis and increasing productivity. Subsequently, Dr. Monica Sharma, also from the same Amity Institute of Information Technology, was talking about the title entitled From Soil to Sustainability, Empowering Agriculture with AI. So, Dr. Monica Sharma was specifically developing sensors. Okay, developing sensors to continuously monitor what's going on in the soil. Because we were also thinking, if we don't monitor continuously, what will happen? But Dr. Monica clearly said her ideas and suggestions. We could also go for a temporal space experiment. Spatio-temporal experiment with this kind of sensor. We can continuously monitor what is happening. It may vary from time to time or region to region. So how to control, how the pH is varying, how the temperature is fluctuating. And how they can implement this AI technology kind of things. Dr. Monica Sharma was presented very nicely. Then eventually, Professor Nagendra Kumar Singh is a BP Paul Chair from a renowned scientist, and he was a director of National Institute of Biotechnology in the ICR system, and he was talking about the genome sequencing. What is the importance of developing genome sequencing using next generation sequencing? He was comparing the Sankar sequence and also the next generation sequencing platform like Solexa and also Illumina kind of sequencing, how the sequencing is advancing the agriculture by deciphering the alleles, specific alleles against biotic and abiotic stress tolerance. And he also emphasized the importance of wild relative collections. It was in fact very curious. He himself collected wild accessions throughout the India, more than 80 rice, wild Oryza glaberrima, Oryza bharati kind of rice. It is wonderful job, and he was presenting how they can be utilized in these generations, how we can utilize along with the genome editing technology, in addition to the classical genetics and breeding, how biotechnology can improve nutritional and stress-related traits, aiming to produce climate-smart crops. That was a nice lecture. So subsequently, On the fourth day, there were 5 speakers were presenting with a theme entitled "Recent Advancements in Genomics and Genome Engineering for Climate Smart Agriculture." Of course, Dr. Professor Rupesh Deshmukh from Central University of Haryana has a lot of, experience in this field. He was talking about aquaporin gene family, especially concerned on NAP group of aquaporin genes, and how there is a competition between silicon absorption and also arsenic, how genome editing can modulate some kind of aquaporin genes. They can selectively allow more silicon and desired metals instead of the arsenic kind of unwanted metals. That is a wonderful work that we need for the modern world to overcome this arsenic, cadmium, chromium, whatever this kind of heavy metal stresses that is he was presenting. Then eventually Dr. Riaz Rao Mir was previously working in Sher Kashmir University, and he recently, few years ago, joined with the group of Dr. Raju Vashne. He was working in various aspects, including, as far as I know, he was working in zinc, also ferrous biofortifications. Nowadays, he's working on nitrogen use efficiency and resilience to heat and drought tolerant in Australia. We have to see, in Australia is one of the country which has affected severely with salt. Okay, yeah, so we have to develop salt tolerant or drought tolerant variety one side, and also we need to increase the productivity upon increasing nitrogen use efficiency. In that way, his research was focusing, and he also tested many populations, some varieties across the environment. Because some genotypes may perform in one environment, may not perform in another environment, and their goal is really good. They are also testing the heritability and across the environment about this population. So that was very nice. Subsequently, Dr. Dinesh Kumar, he was presenting a wonderful lecture about organic farming that can be directly implemented to farmers. We are always talking about gene editing, genetic, these things, but farmers, they may not understand much. So Dr. Dinesh Kumar lecture will be directly implemented to the farmers. They can learn a lot of things. Organic farming is a pathway to climate resilient agriculture, and he was providing a lot of his experience, how we can using vermicompost, organic manure, and intercropping, relay cropping. So many practices he's doing himself along with his research team in IRI, and he was providing such a nice lecture that can be directly implemented to farmers. That's good. And subsequently, Dr. Saloni Mathur also playing the importance of non-coding RNA. The interplay between non-coding RNA and stress responsive pathways for, of course, previous day, Dr. Mishra was also talking similar topic. Similarly, Dr. Saloni Mathur was talking about this non-coding RNA and how the molecular chaperones also, they are controlling the protein modulations and how their interaction with non-coding RNA, microRNA, siRNA, and how they use this efficiently to increase the climatic resilience, especially concerned on drought, heat, salinity tolerance, kind of biotic stress, etc. That was a nice lecture. And subsequently, Dr. Prashant Vikram, as a senior, he's working as a senior scientist in seed science and technology. So he was presenting from IRI about genomics-driven climate-smart crop enhancement prospects, hurdles, horizons. So he also compared the drawbacks also. So he was taking a major green revolution gene, semi-dwarf 1 gene, and how we can improve the using semi-dwarf 1 gene kind of alleles, specific potential alleles to improve the yield without disturbing other important co-segregating genes. He was also talking specifically the Swarna was a green revolution rice variety. He was talking about that with the sub-1 locus. Sub-1 locus is responsible for submergent tolerance, because rice you can also give water, stagnant water, and it will still withstand due to the reason of sub-1 submergence tolerance one. That was wonderful. And upon the use of introgressions from wild relatives, how the SNPs, interspecific SNPs can be transferred, potential interspecific SNPs can be transferred from wild relative to modern cultivars. Even he was talking about Mexican land races, Iranian land races, how we could use it for Indian food security and global food security. That was really a wonderful lecture. So subsequently on the fourth last day, that means today, the speakers, eminent speakers, Dr. Velu Govindan is a leading breeder from the Global Wheat Program, was talking about the zinc biofortification. Not only he is specialized in zinc biofortification, but he also nowadays, CIMMYT whole team also concerned, not only zinc, how to increase yield, because they always yield trade-off. When you increase zinc, they may be yield may be reduced sometimes. So we have to overcome this problem. On the other hand, also how to increase biotic stress tolerance, abiotic stress tolerance. Biotic stress tolerance specifically rust. They can emerge new strains. Periodically they emerge. So how QTL mapping, how genomic selection, how speed breeding and shuttle breeding, how they are doing in Toluca, whole CIMMYT, and how they are coordinating in the ICR system also, how they are taking part along with India to release specific zinc-rich varieties that also having importance for yield. To overcome zinc-related malnutrition in South Asia, not only in India, also they are releasing in our neighboring countries also. So that is a good lecture from him. Then subsequently, Dr. Baines, he has a vast knowledge because he was a former director. He was also associated with the CIMMYT, Borlaug Institute of South Asia, BISA. And he was really telling and asking about a lot of questions. Why? And what is the need? Which gene we have to focus? What is the ultimate goal of us? Just how to incorporate various field, including applied sciences and all. And he was really talking about the genomics, what kind of even genome editing technology and all. He was telling how to use this efficiently. So this with this lectures, I think hope audience getting a lot of benefits about with this 5 days lecture. So please keep on reading and keep on encouraging us. So we, in the future also we can try to make such a program with national and international collaborative speakers. Thank you so much.
Thank you, sir, for that comprehensive overview. Next, I would request the chairperson of this faculty development program, Dr. Doily Sanyal ma'am, to kindly address and share her insights.
Thank you, Deepika. At the beginning, I must congratulate Dr. Karthikeyan for the wonderful comprehension of all 5 days. For someone like me who could not attend due to administrative hurdles and preoccupations, meetings, etc., how wonderfully you spoke what others spoke about, and that shows that you have really heard them well, and it's for the benefit of all.
Thank you.
That
Thank you so much.
yes, people should be attentive while because more importantly, why I'm saying people tend to just switch on the just a moment. I think there are two I'm not able to see. Sorry about that. Are you able to hear now?
Yes,
So,
yeah.
yeah, in a way, people tend to just sleep or tend to go for other occupations or work. I know the issues. I know these things will happen if you are in office, but when it is luxury if you are able to listen to every speaker. So I can see Karthikeyan has done so. Congratulations for that. Anyways, may I wish everyone happy World Environment Day today. 5th June is the World Environment Day, and it's a very opportune day that we are concluding and we are conducting this FDP. So as we conclude this 5-day faculty program, on such a significant day, on behalf of Amity Institute of Organic Agriculture, Amity University, I would like to express my heartfelt gratitude to all those who have contributed to its success. Over the past 5 days, we have had the privilege of learning from eminent scientists, academicians, experts from leading national, international institutions. So the program provided valuable insights into climate change, agriculture, climate smart technologies, artificial intelligence, genomics, precision agriculture, soil and water management, bioresources, policy frameworks, and socially inclusive approaches for sustainable development. Now, the discussions reinforced the importance of integrating science, technology, policy, and community participation to address challenges posed by climate change and achieve resilient agriculture systems. One of the most encouraging aspects of this FDP has been the enthusiastic participation from diverse groups and universities, research institutes, government organizations, industries across the country. This diverse participation demonstrates that growing commitment of the academic and scientific community towards sustainable and climate resilient agriculture. I'm confident that the knowledge you've gained during these sessions will not remain confined to classrooms or laboratories, but will translate into a meaningful research initiatives, collaborative projects, recommendations for policy, and field level interventions. As Dr. Enrico was pointing out, it has to ultimately reach the farmers. Right? On behalf of Amity Institute of Organic Agriculture, I extend my sincere gratitude to all our distinguished resource persons who generously shared their expertise and experiences. As we conclude, I encourage all participants to continue the networks and collaborations that have been established during these 5 days. The challenges of climate change are complex and require collective efforts, interdisciplinary thinking, continuous learning. Together, we can contribute towards building climate resilient agricultural systems that support food security, environmental sustainability, farmer prosperity, and the vision of a developed and sustainable India. I thank each one of you for your active participation and valuable contributions. I wish you all the success in your future academic, research, and professional endeavors. Thank you so much.
Thank you, ma'am, for your encouraging and inspiring words. Next, I would like to request convener of this faculty development program, Dr. Devendra Kumar Chaudhary, sir, to kindly deliver his remarks.
I will be very concise over here, Deepika. Shortage of time. What a wonderful program that we lead over here by keeping the view of cellular and molecular approaches. And certainly, I will use two more words, ex situ and in situ learning. So let's come together for capacity building, and we shall develop in futuristic associations for the worthwhile approaches. Thank you so much. Please go ahead, Deepika.
Thank you, sir. As we now move towards the ending of this session, I would like to request co-convener Dr. Chitranjan Kumar Sharma sir to kindly present his vote of thanks.
Myself, Dr. Chitranjan Kumar Sharma, is Associate Professor at Amity Institute of Organic Agriculture, Amity University. A coordinator for this FDP program, I have been given a challenging task and a worthy task to propose a vote of thanks for the 5 days faculty development program on climate resilient agriculture and allied technology for net zero and socially inclusive framework organized by Amity Institute of Organic Agriculture under the aegis of Amity University, Uttar Pradesh, Noida. Distinguished guests, esteemed guest of honor, respected member of Amity leadership, eminent speakers, galaxy of intellectuals, and vibrant participants, a very pleasant afternoon to all of you. As we draw the curtain on this intensive, enriching, and highly successful 5-day faculty development program, I stand before you with a deep sense of gratitude to propose this vote of thanks. Over the past 30 hours, this FDP has served as a powerhouse of ideas, driving us closer to the nation's visionary goal of Viksit Bharat 2047. First and foremost, we express our profound and heartfelt gratitude to our Honorable Founder President, Professor Dr. Ashok K. Chauhan, sir. Your unparalleled vision, boundless energy, commitment to global academic excellence are the driving forces behind the establishment of Amity Institute of Organic Agriculture and the Amity University. We extend our deep reverence and thanks to our respected Chancellor, sir, Dr. Atul Chauhan and Dr. Asim Chauhan for their exemplary leadership, constant encouragement, and for providing a world-class institution that fosters such high-impact intellectuals across the globe. Our sincere thanks goes to respected Vice Chancellor, Professor Dr. Balwinder Shukla ma'am, whose academic leadership, meticulous planning, and unwavering support continue to steer Amity University, Uttar Pradesh towards global benchmark for research and innovation. We extend our gratitude to the respected Pro-Vice Chancellors of Amity University for their constant institutional guidance. We pay gratitude to the pillars of this program, Professor Dr. Nutan Kaushik ma'am, Director General, Amity Food, Agriculture and Foundation, serving as our esteemed advisor and convener. Thank you, ma'am, for your visionary guidance and structural insights. Professor Dr. Dolly Sanyal ma'am, Director, Amity Institute of Organic Agriculture, our chairperson and convener. Thank you, ma'am, for your transformative guidance, leadership, constant execution, tracking, and for nurturing an environment of absolute academic rigor of Amity Institute of Organic Agriculture. Professor Dr. Devendra Kumar Chaudhary, sir, Amity Institute of Organic Agriculture, convener of this faculty development program, for your painstaking efforts in curating a galaxy of speakers which have made this event an absolute trumps. We highly appreciate and thank our dedicated faculty development program coordinators, Dr. Kartikeya Tyagarajan, Dr. Deepsikha Thakur, Dr. Payal Chakrabarti, Amity Institute of Organic Agriculture. Your round-the-clock coordination, technical management, and flawless execution have ensured this 5-day online program proceeded without a single glitch. We place on record our sincere appreciation to our distinguished faculty members who added immense value to different sessions and moderations. Dr. Ajay Tomar, Dr. Kshitij Parmar, Dr. Deepika Sharma, Dr. Tarun Sharma, Dr. Nikhil Chandel, Dr. Chaitra GB, Dr. Garima Thakur, Dr. Neetu Singh, and all other esteemed faculty members of this domain. Thank you for your collegial support and dedication. The true intellectual of this faculty development program lay in its sessions, and we are heartily indebted to our global icons and national experts from premier organizations like ICAR, NVPGR, NABI, NAVI, ERI, SIMMIT, who graced our digital platform. We thank our distinct galaxy of speakers across 5 thematic areas. Number 1, climate resilient agriculture. Professor Dr. Enrico Porcedu, Chair of the Agriculture Committee, Lindsay, Italy, a world-renowned agricultural genetist who led the establishment of CNR Germplasm Institute in Bari. We thank him for setting a stellar global stage with your talk and several interactions on the food and nutritional security and safety of agricultural production. We thank Professor Dr. Birinchi K. Sharma from Banaras Hindu University for highlighting the vital role of plant microbe synergy in climate resilient crop production. We thank Dr. B.P. Mallikarjuna Swamy, Director of Research, Binhi, Philippines, for presenting groundbreaking progress in breeding healthier rice varieties and biofortification. We thank Dr. Patrizia Gallifi, Senior Scientist, La Sapienza University, Rome, Italy, for illuminating the future horizon of plant functional genomics in climate-smart agriculture. On day 2, we thank Dr. H.B. Singh, Chairman, ICAR, Nanotechnology in Agriculture, for your masterclass on mapping soil-borne diseases through microbial pesticides. We thank Dr. Siddharth Tiwari, Scientist F, NABI, for simplifying the complexities of precision genomic editing, CRISPR-Cas9, and biofortification. We thank Dr. Deva Bandya Mohapatra, Principal Scientist, ICAR, for linking climate resilient to the post-harvest sector through grain value addition and a smart storage system. We thank Dr. Arun Kumar Joshi, former SIMMIT Asia representative, Managing Director, VISA, SIMMIT, for your inspiring lecture on empowering our farmers using climate resilient technological interventions. We thank Professor Dr. Bhupinder Giri, University of Delhi, for your excellent insights into shielding plants from environmental stress using microbial inoculants. On day 3, we thank Dr. Neetu Sanan Misra, Group Leader, ICGEB, for showing her how miRNA act as cellular drivers to rewire stress responses in crops. We thank Dr. Akash Shah, Amit University, Noida, for exploring the transformative capabilities of AI database in modern farming. We thank Dr. Monica Sharma, Amit University, Noida, for impactful session on empowering agriculture from soil to sustainability using AI models and sensors. Professor Nagendra Kumar Singh, President of the Genomics Foundation, for your invaluable session on next generation genome sequencing to target nutritional and stress-related plants. On day 4, we thank Professor Dr. Rupesh Deshmukh, Head Central University of Haryana, for your intricate explanation of aquaporin solute, specifically to engineer safer crops. We thank Dr. Riazul Rauf Mir, Murdoch University, Australia, for bringing global perspective on genomics, assisted breeding, and milestones work, especially from University of Western Australia. Professor Dr. Dinesh Kumar Singh, Principal Scientist, ICAR, for laying out organic farming pathway as sustainable mechanism for long-term climate resilience. We thank Dr. Saloni Mathur, Scientist 5, NIPGR, detailing the regulatory role of microRNAs in climate-smart plant adaptation. We thank Dr. Prasant Bikram, Senior Scientist, ICAR, for highlighting the prospects, hurdles, and horizons of genomics-driven crop improvement. On the last day 5, we thank Dr. Velu Govindan, Principal Scientist Head, Bred Wheat Breeding, Summit, Mexico, for your profound valedictory day presentation on wheat breeding for global calorific and nutritional security. Professor Dr. Navtej Bains, former Director, Punjab Agriculture University, Ludhiana, for mapping out a strategic genetics framework for developing climate-smart agriculture. Thank you to all our session chairpersons who moderated these technical sessions with absolute grace and academic excellence. We thank you deeply. An online FDP spanning 5 days and 30 hours require invisible, robust backbone. We express our sincere gratitude to Professor Dr. Sodhi Sir, Mr. Kapil Solanki, and the entire IT support team. Thank you for your relentless, seamless, and successful digital coverage, Zoom portal management, and real-time troubleshooting that made this virtual gathering feel completely cohesive. Last but certainly not the least, a program is only as good as its audience. We extend our warmest thanks to all our registered participants, the faculty members, research scientists, PhD and PG scholars, industry experts, and NGO professionals who joined us from across the country and overseas. Thank you, your valuable presence, your active interaction in question answer sessions, and your commitment to completing the rigorous daily assessment. We hope the knowledge gained here will successfully translate into respective classrooms, laboratories, and fields. I also wish to thank all experts and others whose names I have forgotten to mention. Forgive me. As we leave this forum, let us carry towards the spirit of sustainability, net zero commitment, and social inclusivity that this FDP has instilled in us. Thank you once again, everyone. Have a wonderful evening ahead. Best of luck. Jai Hind.
Thank you, sir. As we reach the conclusion of this valedictory session, I would like to express my heartfelt appreciation and gratitude to all the resource persons and all the participants for their enthusiasm and dedication throughout the program. With these words, we formally conclude the valedictory session of this faculty development program. Wishing you all continued success in your academic and professional endeavors, and best wishes from Amity Institute of Organic Agriculture, Amity University, Uttar Pradesh, Noida. Thank you.
Deepika, may I take a moment to reiterate that certificates will be issued online to their respective email IDs, provided they have taken the test and secured a good percentage and have attended more than 90% of the lectures.
Right.
So please wait for us to evaluate these two conditions which we have put, and then your certificates will be sent to you in due course. Thank you all. Have a great day, and of course, Environment Day. So keep the environment safe and play the crucial role as an educator. Bye-bye.
Bye. Bye to
Thank you.
all. See you in near future. Bye bye.
Thank you, sir.